This lecture covers the second part of Chapter 7, the muscular system. It contains muscular movement at the cellular level. So let's start with the functional unit of the muscle. Skeletal muscle is divided up into several layers of cylinders packed inside each other and covered in sheets of connective tissue. We're going to start with the smallest unit and work our way to the largest. So myofibrils are rod-shaped organelles within the muscle cell, also known as the muscle fiber. The muscle fiber, or the muscle cell, is up to 12 inches in length, and it is made up of myofibrils. Endomysium is connective tissue which surrounds each muscle fiber. Bundles of muscle fibers are held together by the perimysium to form fascicles. Bundles of fascicles are held together by the epimysium, which is continuous with the muscle tendon. So to review, the tendon turns into the epimysium, which covers the fascicles, which are surrounded by the perimysium. Each fascicle is a bundle of muscle fibers, or muscle cells, covered in endomysium, and is a bundle of myofibril. Myofibrils are cable-like strands of protein strands called sarcomeres. It is the sarcomere which allows the muscle to contract. So sarcomeres are contractile units of muscle with two types of protein threads called myofilaments. Thick filaments are made up of myosin, and they contain myosin heads. Thin filaments are made of actin. The actin and the myosin arrange themselves in layers between two Z-lines. This forms the contractile unit called the sarcomere. So you can see here that the actin and the myosin are sandwiched in layers between two Z-lines. Repeating sarcomeres are what give muscles striped or striated appearance. During contraction, temporary attachments form between the myosin head and the actin, and these are called a cross bridge. The myosin then rotates and pulls the sarcomere together. So you can see here that the Z-lines have come closer together. When the contraction is over, the cross bridges release and the sarcomeres lengthen again. This is a really small change, but imagine it happening in hundreds of millions of sarcomeres at the same time. It is the shortening of the sarcomeres which allow for contraction. Muscle contraction starts with a neuron which contains the neurotransmitter acetylcholine. This neuron hovers over the cell membrane, also known as the sarcolemma. The sarcolemma contains many sodium ion channels. Within the cell, we have the sarcomere, which is made up of the thin filament actin and the thick filament myosin. Notice that the binding sites on the actin are currently covered up. Within the muscle cell, there's an organelle that is similar to the endoplasmic reticulum called the sarcoplasmic reticulum. It's filled with calcium ions. When the neuron releases the acetylcholine, it binds to the sodium channel, causing it to open. Sodium then floods into the cell, causing the sarcoplasmic reticulum to release calcium into the cell. The calcium then binds to the protein on the actin, lifting the protein and exposing the myosin binding sites. The myosin is now free to bind to the actin. ATP then binds to the myosin and causes it to change shape, pulling the heads backward and shortening the sarcomere. ATP loses a phosphate and becomes the less energy-rich ADP. This breaks the bond between the actin and the myosin. Sodium then floods back out of the cell, causing the calcium to flood back into the sarcoplasmic reticulum and the binding sites on the actin become covered again.